I don't realize if, is this sounding like a microphone? Okay. So thank you very much for staying after a very long day. I will try to combine a bunch of things because as well, we've been telling you for a while now that where? Hola? Okay, okay. There. Okay, fine. So let me see if it doesn't fall over there. Okay, so we are trying to see if we can converge into some network of biological physicists, and also if we can combine our expertise to work on something. And not everybody could come here, so what I will try to summarize here is, on one hand, based on my own experience, what type of contributions physicists can make to try to answer some biological questions, and then describe some groups that are not represented here from Argentina, and finally give you some ideas of some networks that already exist that could serve as inspiration for whatever network or project we want to define. And those are going to be the discussions on Sunday. So if we want to form a network, what type of network, what kind of funding we can get for that, whether we want to define a joint project, and then once we finish the discussion, define some action plan and assign tasks. So please be present for the task assignment. Anyway, so one question is, are we biological physics or biophysics? And so biological physics was coined basically to sort of distinguish the group of physicists who started to respond, to give answers to some questions of biological origin. It also refers to biological inspired physics. And so it was going to distinguish it from the usual biophysics word that refer to people who has been trained in other areas. It doesn't mean that we don't collaborate, but I think it was a coined for that. And it's an area that began to grow with a Human Genome Project. And it was at that time, in the 90s, that many physicists started to move into biology. And so that's sort of my story, too, that I moved from doing research on dynamical systems mostly, and also known equilibrium physics, and try to look at biological systems. And I have some collaborators who are biologists working on systems biology, and one of them says, oh, biological physics is the same as systems biology. But anyway, if you Google systems biology, according to the Google AI, it says it's an interdisciplinary approach that aims to understand complex biological systems by studying the interactions between their components, using computational and mathematical models, and integrating data from various fields, which is something that many biological physicists do, I would say. Anyway, so this is an example. I will try to be brief. It's an example in which I worked on. For the last many years, I worked on calcium, intracellular calcium signals. Calcium is a universal messenger. It is involved in many processes. One of them is fertilization. That's a fertilization wave in a Xenopus laevis egg. And what you see white there, that wave traveling there, it's a calcium wave. This is where the sperm anchors, and then it launches this wave, and that starts all the process of enzyme activation that is necessary for the cell division. It's involved in many other processes, including neuronal communication, as we saw with those of you who were here for Olga's talks, for the vesicle fusion and release of neurotransmitters. And it's also involved in muscle contraction. And so one of the questions that we were puzzled with was how it could be universal and specific. And one of the answers is that depending on how it's distributed in space and time inside the cell, it elicits different responses. And so in the case of, of this wave, that calcium is inside the endoplasmic reticulum and is released from the endoplasmic reticulum into the cytosol through channels that are called IP3 receptors. And so that distribution, the resulting distribution of intracellular calcium, depends on how those channels are distributed in space. The fact that those channels, their open probability depends on calcium, and that endows the cytosol with some excitable property, and many other aspects. And this is about the channels that I just talked. And the resulting propagation can be saltatory, which means that you can observe the individual release sites because you release calcium and you see that local elevation, then calcium diffuses, gets into some other region where there is another channel whose open probability increases in the presence of calcium. So now it becomes open, releases calcium, and so on. And so you can have this saltatory kind of propagation, or you can have something more continuous like the one you see there for fertilization. And the saltatory propagation can also fail to propagate because it's like you no longer have this excitable thing if the release sites are too far away from one another. And so we decided to study that transition in a system which is the oocytes also, the immature oocyte and the the egg that can be fertilized. And because in that transition with maturation, there is a spatial distribution of the channels through which calcium flows into the cytosol. And so these were experiments performed by a former student of mine, Stefania Piegari. And so what we did was used something to induce the release of calcium from these channels. So we sent UV light to open the, to put something that would open the channels, basically. And this is the immature egg, and you saw those spots. We submit, and then you see something more continuous, but you see the individual releases. And the other one is the mature egg, and what you see is something more uniform. And so we thought that the underlying spatial distribution that was different of the channels was the cause of this different type of propagation. We elicited, we sent 2 UV flashes. That's why we see 2 elevations of calcium. This is for the mature egg, and then we did that for the mature egg, so the mature oocyte and the mature egg. And we saw differences in the way that calcium was released in both. But then when we did a very simple modeling with clusters of channels and a very simple way of inducing the calcium release in the model, we found the distributions of the types of events, if the events were more localized, so that the sites of release were more distinguishable, or if they were more continuous, then the key factor to switch from one type of release to the other was not the spatial distribution of the channels, but it was the rate at which calcium is removed from the cytosol. And during maturation, there is also an invagination of the pumps that take calcium out of the cell. So through a very simple model, we were able to understand that transition from the mature oocyte to the mature egg was not just because of the redistribution of the calcium channels, but it was mainly due to differences in calcium removal. So that was something that we thought it was interesting, an interesting contribution combining the experiments and the modeling in a physics style. Anyway, so I am a professor at the University of Buenos Aires, which is very big, but in particular, I'm a professor at the same school as Diego is, which is the School of Exact and Natural Sciences. These are the buildings. And it's very particular and very interesting because the university is spread all over the city But in this school, we have all natural sciences, all STEM areas, but engineering and technology is not there. But we have physics, mathematics, computer science, biology, etc. So it's a place for interdisciplinary research. It has many researchers. It has many graduate students. These are some numbers of our school. And there are several research efforts in cross-disciplinary areas in physics and biology. And so there are too many. Diego is, I would say, is one of them, of those examples. But I'm going just to list those that are led by physicists, just because to narrow the thing. So there is an institute called Instituto de Cálculo. There we have Luciana Bruno, who works on mechanical interactions between mitochondria and the cytoskeleton, AI tools for the automated analysis of cytological images of pap smears. And so I listed some recent papers so that you could look at their titles. And then Guillermo Solovey, who recently moved out, but momentarily, from our school, and he does cognitive science, statistics, data science. And then in the Department of Computer Science, again, you have a physicist doing brain activity in natural situations, neurophysiology, computational models, and clinical applications. And then Edgar Altsiller, he does more complex systems, I would say, but he also does AI applications to study behavior, social psychology, and mental health. So here you see like many doing things related to neuroscience and image analysis, and Luciana does more like transport inside the cell and uses a lot of microscopic experiments for that she analyzes. Then these are other institutes with physicists leading some groups. Alejandra Ventura, she's a former student of mine also. She does cell signaling, information processing in biology. In the same institute, there are two other women physicists, Luz Babasi, who does neuroscience and memory, and Veronica Perez-Schuster, she's also working on neuroscience. She's less senior, I would say. And then this is people who are professors in our school, but their research labs are not inside our school, but they are in nearby institutes. One of them is Luis Morelli, and he does information processing in cells also, and tissues. And in Instituto Leloir, we have Emilio Kropf, who works on brain algorithms for spatial orientation and memory, physiology, modeling, and applications. He works on how mice navigate the space and the grid cells that are associated to different regions of the space that is visited by the mice. So all of them, well, many of them, this is theory or modeling. Luis is also modeling. Luz and Veronica, they do experiments, and Emilio does a mixture. Oops, something happened. It's not, let me see. Maybe I touched something. Type, it says something. Okay, there. So I'm going to move out of Buenos Aires to Cordoba. I think that there is a student here from Cordoba, but good. But not senior researchers or professors came this time. So I'm going to list a little bit. Carlos Condat, who does mathematical modeling of oncological processes mainly. He's already retired, but he's continuing to do research. Gustavo Sibona, who does also modeling and simulation of biological systems. Veronica Marconi, she works on soft matter and does microfluidics, lab on a chip, and colloids. They are all modeling people, or they do analysis of experiments. And Lucas Barbieris, I think, is your advisor. And he does modeling of biological systems by means of computational tools and looks at movement of different types of animals. And, well, I don't remember your name, but there was a nice poster with with your work there. So, and then let me go back to the suburbs of Buenos Aires. Universidad Nacional de General Sarmiento is Florencia Carusela. She works on also transport inside the cell, and although she also does some applications, more technologically oriented applications of thermal transport. And Alejandro Monastra, who also does transport and collaborates with Luciana Bruno. At Universidad Nacional de General Sarmiento, there was a group that now one of the professors died, another retired. That was a node of an interesting network that was called Infernet. You know that Fernet is a type of beverage that is very common in Córdoba, and which was a network of people doing mostly bioinformatics and network science with collaborations like organized around former students and postdocs of Giorgio Parisi in Roma. So many people from Italy in that network. And I don't think it continues anyway. So then Universidad de San Martín, we have Dante Chialvo, who was mentioned by Tomás, I think, in some of his projects. And Julián Ayadi, Julián, who does microscopic techniques. He's truly experimental, and he's more on microscopy, not just for biology, because, well, he does modeling. Oops, I forgot. I didn't. That's not the thing. I mean, that's not his theme. The theme is wrong, but the papers are right. So those are two papers he recently wrote with biological applications of the microscopic tools that he uses. So he's an experimentalist. So several physicists, they work on microscopy, some of them on photonics, and applied to biology. One of the largest groups led by a person who is a professor of the physics department in Buenos Aires is Fernando Stefani. He does optical manipulation and characterization of nanoscopic systems and super resolution fluorescence microscopy. He develops very beautiful new techniques to observe things at very, very small scales. Some of them applied to biology. And I wanted to mention a very interesting network. I expected someone from this network to come and talk about it, which is LABI, Latin American Bioimaging. It's a network of imaging scientists that are interested in improving training, education, and access to imaging technologies across Latin America and the Caribbean. And so now I'm going to move from the list of some people in Argentina that are not working at the physics department, where I am based, because Ana Amador from the group of Gabriel Mindlin, she's going to talk about people in the physics department tomorrow. And now I'm going to move to these networks, examples of networks that could be used also as an example of what we could try to think of implementing. And the actions of LABI include building community and building capacities via the organization of annual meetings, satellite events, and they also have working groups. They also have training and career development programs. They give travel grants to users, to researchers, to technicians, to attend courses, and to establish collaborations. And also they want to share useful tools, like, for example, tools to analyze images and stuff. And one of the ideas is also to share equipment so that people can go to another lab to perform their own experiments, so that you don't need to have your own lab set up. And this is drew some inspiration from something that is called global imaging. And this idea of sharing equipment resonates with other initiatives, some regional, and some, I'm going to mention one regional initiative and one from Argentina that is no longer being funded, but it's there. One is this Latin America Strategy Forum for Research Infrastructure, Last for Free. this started with the director of the ICTP in Trieste, Fernando Quevedo. And so since he is from high energy physics, the idea initially was to try to discuss what type of shared facilities could be built in the region, or what kind of equipment, or try to combine the infrastructure for high energy physics in the region. I know that Rogerio Rosenfeld, who was director of this institute, he was also very much involved into that. And they drew inspiration from something similar, but with much more funding, of course, in Europe, which is called ESFRI. And then in Argentina, although now with no funding, we have what is called National Systems Program. That was launched by the Ministry of Science several years ago. And so the idea is more or less the same as how it's defined by ESFRI. Basically, this research infrastructure are facilities that provide resources and services for the research communities to conduct research and foster innovation in their fields. And they include research laboratories, scientific instrumentation, technologies, data resources, computational tools, communication networks, and they are open and accessible. And in the case of Argentina, the national systems was created for the efficient use of physical resources for the STEM communities, the productive sector, and society. And as of July 2020, there were 12 systems. For example, there was one that is called of microscopy, and you can have your lab associated with the program, with the national system. My microscopy, these experiments that we did in all sites and stuff is in a lab that I am the responsible for that lab. And so we associated that lab with the national system for microscopy. That means that we have to offer that 20% of the time be offered for the use of the community, whoever wants to use it. Of course, we charge for that. There is a fee. We need a technician to operate when we have visitors coming to use the microscope. And then you are allowed to ask for improvement, for updates of your equipment. So it's so that not everybody has the same equipment, but you put a large equipment in a particular place. They offer services for the community so that the community could come. And so a LABI has that idea too. But of course, at the international, a regional level, it's much more complicated because it's much more expensive to move around. But we can also think of if samples could be shipped, that technicians could perform the experiments. And even there are also efforts, particularly, I know in Africa, there are efforts for the long-distance manipulation of laboratory equipment. There are many things that you can think of to help regions that are less developed be able to access a very good facilities at the experimental level. Now the biophysics community in the region put together this LABI, and they have a long tradition of collaboration, and I think we can get inspiration from that. Particularly, they have this post-LATAM program that was started by the father of Tomás Grigera, and now it runs under the auspices of LAFEPS, which is the Latin American Federation of Biophysical Societies. So basically, the way that I think this is, or I describe it for myself, some sort of umbrella organization, the post-LATAM, that signs agreements with postgraduate programs that already exist in universities of our region. And so that it was done with, so that people do their PhDs or their master thesis in those institutions, but then if they are approved by the program, they receive like a stamp that they received a postgraduate degree in biophysics in Latin America or something like that. And so post-LATAM controls the quality of the courses, the quality of the thesis, but also they organize, I don't know if they have much funding now, but at the beginning they used to organize many courses with full travel grants for students to go and attend those courses. So the idea was to help some universities that didn't have that much, I mean, they were not so developed, but also to favor interdisciplinary research, because maybe you were doing your PhD in physics or in biology or in chemistry, but in some interdisciplinary type of theme, and so then you could like receive some stamp that was about that your work was interdisciplinary. Anyway, so our subject is highly interdisciplinary. Even if I started trying to define a subset of people with physics training, I think that it's very important to collaborate across fields. And for that, this I kept in Spanish, which is we need to listen to other people and learn the language of the other people, learn to communicate. And this diversity and inclusivity is not just be able to work across fields, but it's also very important to be inclusive in many other aspects. Particular, one of them is gender, because science that is more inclusive is a science that is more humane, and that's a better science. So with this, I leave you.
Thanks, Silvina, for this nice presentation. We have some time for some questions.
Thank you, Silvina, for the very broad view of how is biological physics in Argentina now. I wonder if you thought about involving CLAF in this initiative, the Centro Latino-Americano de Física, which is based in Rio de Janeiro. So I was in the board of CLAF a long time ago, and I think that's the kind of project that if I was there, I would love to receive, because then you can somehow help funding and increment all the interactions between different groups in Latin America. And also, I think CLAF can access funds in other instances like UNESCO, I don't know. So my suggestion is to knock the door there.
Yeah, well, CLAF, I think it's a very, very important tool that we have in our region, but unfortunately, the countries are not paying their dues. I mean, I think that it's mostly funded by Brazil. Other than Brazil, the rest of the countries, I think they are, way behind they are not paying. So, plus, the last director from Chile, he didn't even move to Brazil, and so it was kind of getting a little, not very active, I would say. But I hope that with the new director who is based in Brazil, things will change. UNESCO doesn't put any money. I mean, in the ICTP of Trieste, they don't put any money. They put their name and their rules and their prestige. That's UNESCO. But I think that CLAF used to organize a school of physics in the 60s and 70s, I think. And that created a community which I would like very much to recreate, maybe not of physics, but maybe of biological physics or something that we can collaborate. And it would be a super important tool, but I guess they could collaborate with the ICTP that has much more funding. And they have these beautiful premises where we can organize the schools, but we should get them involved for sure.
I participated from this creation of LAFESB, and I was president of the Brazilian Biophysical Society. And when we have created that, we have the support of the International Union of Biophysics. The International Union of Physics has a section named Biological Physics. I don't know, there are someone in Brazil, I think
I am the president of IUPAP now, of the International Union of Pure and Applied Physics. Yes, I am the president. There is a commission on biological physics.
Yes, yeah, I have participated also in this commission.
And so they are involved in the organization of a large international conference. We have some representative from IUPAB, the biophysics there, but we are not funding projects or research, and we don't have that much funding to be able to do that. But we are discussing what to do to increase our impact. So that's a possibility.
Yes, but LAFESB was created as an organization, and after we have looked for support from Brazil, we have, I think, also from Argentina somewhere.
Yes, so
because we have we have planned exchange between students from Brazil to the other countries, and somewhere Brazilian Minister of Education will support that.
It would be, on Sunday, we will have the opportunity to discuss all of this. This I wanted to give just like an input for you to know about all these initiatives. And then on Sunday, we can discuss what we can do and where we could get money from.
Okay.
Thank you.
Okay, we have time for one quick question, if any.
Okay.
Okay, so let's thank the speaker.